About the cardiac muscle cells known as cardiomyocytes, or simply cardiac muscle cells. Um, these make up the majority of the heart mass. These are the contractile cells. There are other cell types associated with the heart tissue, right? There are connective tissues. There's neural tissue embedded in the heart muscle. There's right? There's vascular tissue embedded with the heart muscle, but I want to focus on the heart cells that are contracting for the moment, and these are known as cardiomyocytes. And if you think about it, the term is kind of telling you cardio refers to heart, myo refers to muscle, and cyte means cell, C-Y-T-E means cell. So it says heart muscle cell, right? So anyway, what I want to talk about, we have an electro micrograph here, microscope image of cardiac muscle, and it's hard to note, but it is striated muscle, right? This is one of the key characteristics of it, and you probably learned that previously in Anatomy and Physiology 1. It is striated. And striated literally means striped. And again, you can see some of these big stripes. Those aren't the ones we're referring to here. These are called the intercalated discs. But if we had a little bit closer, higher magnification, a little closer view, we would see that there are stripes there called striations. And that is due to the presence of sarcomeres. And we can see some of the elements of the sarcomeres down here with the thick filaments overlapping with the thin filaments. Right? There's the Z line that's helping suspend the thin filaments, right? So we've got the components of the sarcomere sort of laid out here. Again, just to point out, this micrograph, if we follow my arrow here, right? This arrow I'm highlighting in red, right? That's a gap between two adjacent cells. We could actually we could label these cells as A and B if you like, right? With this gap space between them pointed out, and that's going to be called the intercalated disc. The smaller stripes that we would see under higher magnification would be due to these alternating thick and thin filament regions within the sarcomeres that generate a striated appearance. Another characteristic of cardiac muscle, is that these cells are single nucleated. They have a single nucleus, one nucleus. And that might seem like an odd thing to point out. However, when we talked about skeletal muscle tissue is what's known as, the cells themselves are known as a true syncytium, which is, we'll come to syncytia in a minute here. But what that means, a true syncytium, is when cells fuse. And in skeletal muscle, many cells fuse to form the skeletal muscle fibers, which is why those cells can have, in many cases, dozens or even hundreds of individual nuclei. All right, but these cells do not form by cell fusion, so they have a single nucleus. Now, what they do have, in addition to that nucleus, is a very large number of mitochondria. And again, I don't think we can really pick out any mitochondria in our micrograph over there. I've got a couple of them labeled down here. I can highlight, right? This guy is our double membraned mitochondrion. Here's another one over here. All right, I'm giving it sort of a blue outline here. Right? And so those are the mitochondria, and they actually form about 25% of the mass of the cell, right? So it's an enormous mitochondrial volume in these cells. 25% mitochondria. And if you'll recall, the mitochondria is where cells aerobically make ATP. So what it hints at is that these cells make a lot of ATP continuously. And if you think about it, that makes a lot of sense because our heart has to work day and night, right? Whether we're sleeping or not sleeping, our heart is pumping blood. And so it needs to very effectively make ATP to fuel the cross-bridge formation of these thick and thin filaments that we highlighted just a moment ago. Right? And so a very large number of mitochondria and the associated enzymes that would be found there that help with that aerobic ATP production. If you recall, anaerobic ATP production is really inefficient, and heart muscle would not function effectively if it becomes anaerobic, and which is effectively what happens during a heart attack when we lack sufficient blood flow due to a blocked vessel. The heart muscle becomes ischemic, which means it does not have enough oxygen, and because of that, the cells will begin to die. So again, we need a large number of mitochondria to utilize a large amount of oxygen in that ATP production. So another thing that goes along with that would be a very high capillary density. Right? This is a highly aerobic tissue. So having a lot of capillaries closely associated with the outside of these cells, and again, not something you can pick out in the micrograph up here, but they would be there would be a lot of capillaries, which aids both the diffusion of oxygen into the tissue as well as the carbon dioxide and other waste products out of it as well. And we can think about nutrients coming in, essential components, right? So very high capillary density. And the last bit that sort of linked together under the required for high aerobic energy production would be myoglobin. Myoglobin sounds like hemoglobin, right? But myoglobin is an intracellular, well, hemoglobin is too, but it's in red blood cells. Myoglobin is found in slow twitch skeletal muscle and also cardiac muscle. And it's a pigment, a protein that is pigmented, colored, which gives cardiac muscle a darker red color. But what it does is it binds oxygen, right? So this guy is going to bind oxygen and create, we can view it as an energy store within the cells, and that's true to a degree, like sort of extra oxygen on site. But even more so, it probably serves as sort of a middleman helping to convey the oxygen that's diffusing, right? If we want to think about it, we could have oxygen, right, coming from our capillaries, diffusing into these cells, right? And actually, let's have it go towards our mitochondria here, right? We're going to use it. Diffuses everywhere, but diffuses into the mitochondria where it's utilized. Now, the myoglobin will actually bind it as it enters the cell, and then sort of like, if you will, sort of hand it off to the mitochondria. And what it does is it keeps the gradient favorable by keeping the oxygen bound intracellularly, and aids the diffusion, right? So yes, it's an oxygen store, helps avoid the tissue becoming hypoxic, low in oxygen, but even more so, it probably acts as a middleman, if you will, sort of passing oxygen from the cytoplasmic source to the mitochondria. Okay, what other characteristics do we have here? So, so just to summarize though, let's put these three characteristics together under, right, aerobic. Put exclamation point, right? You gotta remain aerobic all the time, make lots of ATP to fuel this continuously functional, active muscle. All right, another key characteristic, one which is pointed out here in the image, right, are these darker stripes, if you will. They're called intercalated discs. And these are found where the cells butt up against one another, right? Remember I said we've got cell A and we've got cell B, and right here they meet, right? This arrow is pointing at this region called the intercalated disc. And so there would be interstitial space there, right? This gap, this sort of white gap in between here. But you'll notice there are proteins that span between the cells. And there are two specific cell-cell junction protein types, or categories, if you will, which are really important, right? So let's list these as a function. So this is intercalated discs. right? And those intercalated discs have two components, gap junctions and desmosomes. Hopefully those terms are a little bit familiar. You should have heard them before. Right? They serve two very distinct functions. Gap junctions, this guy right here, I'm going to highlight it in the bright purple. Right? Gap junctions form pores in the two adjacent cell membranes, and then they sort of dock to form a gap that connects the cytoplasm right between the two cells. So what they do is they effectively join the cell's cytoplasm from cell A to B, and then B to C, and C to D, and so on and so forth. What that means is that these cells are somewhat contiguous in terms of their cytoplasm. And the important element there is that ions can move rapidly across this cytoplasmic bridge through the gap junctions. Right? So gap junctions are about communication. They are about conveying the stimulating, it's a depolarization, the stimulating change in membrane potential that's going to initiate the contractile sequence. Right? So gap junctions are for communication, and that communication is in the form of electrical current passing through them to stimulate one cell, then the next, then the next. So you can think of it like dominoes falling, right? When you push one domino over, they fall sequentially one after another because they're close together. Well, in this case, they're falling because they're literally linked by these gap junctions, right? and the falling over in this case would be contracting. The other cell-cell junction is this series of proteins, excuse me, this series of proteins, sort of highlight them in green here, right, that are sort of connecting the two cells, not in terms of a communication function, but a structural connection, right? Those guys basically link the cells structurally. I'm highlighting the other gap junction down here, right? So two gap junctions and two desmosomes. So the green desmosomes here, right, are the cell-cell junction proteins that tie the cells together mechanically, right? Gap junctions were for communication. Desmosomes were for mechanical connection, right? Gap junctions for communication, desmosomes for mechanical, structural, right? That's what I mean by mechanical, a structural connection. Think about it. What is cardiac muscle pulling on? When it contracts, right? Your skeletal muscles pull on tendons, which pull on bones, right? Most of them, right? There's a couple facial muscles that don't do that, but right? But most of them are pulling on bones. What's the heart pulling on? Is it pulling on your sternum? No. Heart muscle is pulling on heart muscle. It's just simply squeezing to generate pressure inside its chambers to move the blood. And so we can't have, right, because it's the cells are pulling on each other, we need to make sure that they don't pull apart. Desmosomes, right? We need them all to contract in sequence and almost, but not quite, in unison. That's the function of the gap junctions, right? The communication so that the signal can travel rapidly from cell to cell to cell, right? We'll talk about the nodal cells and the conducting system in a separate video, but that stimulus has to be generated, nodal cells, right? But then it needs to travel cell to cell so we stimulate all the cells, right? And they fall like dominoes, all contracting one after another very quickly, right? And very quickly, right? The time separation between the beginning of the heart contracting and the end of the heart contracting within a single beat is a small fraction of a second, right? So these signals travel very quickly. Now we want to put a label on this, right? In other words, these intercalated discs with their gap junctions and desmosomes form something called a functional syncytium. Right? I mentioned a minute ago, I mentioned a true syncytium, right? When I talked about skeletal muscle. True syncytia are formed when cells fuse. A functional syncytia is formed when cells don't fuse, but they act as a unit. And in this scenario, they're acting as a unit because they are mechanically connected, desmosomes, and they're connected for communication, gap junctions, so they can work as a functional syncytium or a functional unit. Okay.